Good evening, everyone. My name is Haejin Cho, and I'm from Food Microbial Genetics Lab from third floor. And today I'm going to give you a presentation about a paper related to my research. The name of this paper is Transcriptome Sequencing Revealed an inhibitory mechanism of Aspergillus flavus asexual development and aflatoxin metabolism by soil fermenting non-aflatoxigenic Aspergillus. So this will be my presentation index. So this paper is usually talking about two types of Aspergillus strains. First one is Aspergillus flavus. This flavus is a common saprophytic fungus that contaminates the crops. So they mainly causes huge economic losses to agricultural industry. This Aspergillus flavus is an opportunistic pathogen for immunocompromised patients. So this is the second most common cause of aspergillosis next to Aspergillus fumigatus. This flavus is a very well-known producer of one of the most carcinogenic mycotoxins, aflatoxin. Next strain is Aspergillus oryzae. This is a filamentous fungi, also known as koji mold, and it is used in the food fermentation industry, and it is generally recognized as safe. This oryzae is usually used in East Asia to make alcoholic beverages such as sake, and also used to ferment soybeans in Korea, beju. Actually, I took this picture in my lab, and as you can see, this flavus and oryzae looks very morphologically very similar. These two fungi are very similar in genome size and number of predictive genes. Previous studies showed that the number of mutations exist in aflatoxin biosynthesis gene cluster in Aspergillus oryzae, which makes it non-aflatoxigenic. So what is aflatoxin? Aflatoxin is mycotoxins that are produced mainly from Aspergillus flavus and parasiticus. This is well known as group 1 carcinogen, and long-term exposure of aflatoxin may cause kidney, liver, lung, or colon cancer in both animals and humans. This aflatoxin naturally occurs in foods such as groundnuts, peanuts, maize, rice, and some dried foods, and it as a result of fungal contamination to food or before and after harvest of maize and kernels. So more than 18 different types of aflatoxins have been discovered these days, but the most common and important ones are aflatoxin B1, B2, G1, and G2. Aflatoxin B1 is the most common in food and among the most potent genotoxic and carcinogenic toxins. So aflatoxin contamination is highly dependent upon temperature and humidity. These two conditions not only encourage the growth of Aspergillus flavus, but also have great effect on the activation of the aflatoxin biosynthesis gene cluster. Numerous strategies, including physical, chemical, and biological control, have been applied to combat this fungus and control aflatoxin production during harvest. Physical control includes heat and ultraviolet radiation treatment, and chemical control is using natural preservative. And last, biological control is using the microbial competition. So biological competition is nowadays, researchers are very focusing on this control. This a range of microbes such as bacteria, yeast, and fungi have been used for biocontrol aflatoxigenic Aspergillus. These have been studied recent days, and these were reported to inhibit aflatoxin production and inhibit growth of Aspergillus flavus. So this paper's researchers wanted to build a system that uses non-aflatoxigenic Aspergillus strains as a biocontrol. So the aim of this study is to use non-aflatoxigenic Aspergillus strains. In this paper, they used grass strain oryzae and a Aspergillus flavus mutant that cannot produce aflatoxin, and cultured together with flavus wild type. They wanted to elucidate the effect of non-aflatoxigenic Aspergillus strains in the production of the aflatoxin B1 and development of flavus in co-culture system, and to reveal the inhibition mechanism of aflatoxin B1 metabolism and flavus development by these non-aflatoxigenic strains. So These are the methods and materials. First, these are the strains that are used in this study. First, the strains that produces aflatoxin was Aspergillus flavus wild type, and the strains that did not produce aflatoxin was Oryzae. We used was Oryzae wild type, and this TSJ1, which is a deletion mutant of Aspergillus flavus that fails to produce aflatoxins. So these strains were all inoculated on potato dextrose agar medium and cultured in these conditions. So this is the method of co-cultivation. So firstly, non-aflatoxigenic Aspergillus strains were incubated, and the spore was then co-cultured with the aflatoxigenic flavus and incubated in these conditions. And after that, aflatoxins were extracted from that co-culture system, and a thin layer chromatography was performed. Finally, culture filtrate of non-aflatoxigenic Aspergillus strains. This is method of making culture filtrate. So first, a total number of non-aflatoxigenic Aspergillus strains were incubated, and the cell-free culture filtrate was collected by removing the hyphae and using this 0.2 micro diameter filter. And the conidia suspension of Aspergillus flavus was added to medium in supplementation of these filtrates and were incubated in these conditions. So these are the results. So firstly, the researchers wanted to know the effect of non-aflatoxigenic Aspergillus strains on Aspergillus flavus growth and aflatoxin B1 production. Firstly, they checked the biomass when they are cultured together, and they recognized that the total biomass didn't change when co-cultured or single cultured. But as you can see, this is the mycelium pellet, and you can see that when they are co-cultured with Aspergillus oryzae, the mycelium pellets were bigger, and when they were co-cultured with non-aflatoxigenic flavus, the pellet was smaller. Also, they wanted to know the effect of co-culture system in aflatoxin B1 production. So they checked the aflatoxin production in increasing amount of conidia. This is when co-cultured with oryzae, and this is when co-cultured with non-aflatoxigenic flavus. And you can see the increasing conidia amount to this conidia apparently blocked the aflatoxin B1 production in the co-culture system. Also, when at both 3 and 9 days of incubation, we can see that co-culture system totally, especially this oryzae co-culture system, inhibited the production of aflatoxin B1. Next, the effect of the culture filtrate of Aspergillus oryzae and this non-aflatoxigenic flavus was measured. So inhibited growth of Aspergillus flavus was found within the treatment of oryzae and supplementation. You can see in this colony, and this is the quantitative analysis. You can see that when co-cultured, they grow smaller compared to wild type, and both same in this TSJ1 strain. Also, the researchers counted the conidia number from each plate, and they found out that when cultured together, the conidia production was inhibited compared to wild type when co-cultured. Also, they checked the aflatoxin B1 production, and you can see that when co-cultured with Aspergillus oryzae, aflatoxin B1 production was inhibited almost 90%. So these results imply that culture filtrates of non-aflatoxigenic Aspergillus have inhibitory effects on Aspergillus flavus conidia production and AFB1 production. Next, the researchers wanted to elucidate the role of co-culture systems in Aspergillus flavus development or aflatoxin B1 production. So they carried out a RNA-seq analysis. Aspergillus flavus grown in the presence or absence of oryzae culture filtrate was further analyzed with using RNA-seq, and the RNA-seq results showed that 3,133 genes were differentially expressed in response to Aspergillus oryzae filtrate supplementation. These are the top 10 upregulated genes, and they were almost 800-fold increased compared to wild type. And these are top 10 downregulated genes, and they were almost 50-fold decreased compared to the wild type. And this, based on the RNA-seq analysis, they analyzed the gene ontology terms, and the most enriched geo terms when supplemented with uridine filtrate was carbohydrate catabolic process, integral component of the membrane, and extracellular region. Also, they ran a KEGG pathway analysis, and they recognized that upregulated GAG differentially expressed genes in the treatment of Aspergillus uridine filtrates were enriched in primary and secondary metabolism. This is too small, so I'll just say it. And downregulated DEGs were significantly enriched in the metabolic pathway, antibiotic synthesis, and the precursors of secondary metabolites. So they are mainly involved in metabolism. So the synthesis of secondary metabolism in Aspergillus flavus might be inhibited with the treatment of uridine filtrates. So then they wanted to know the effect of Aspergillus uridine filtrate supplementation in aflatoxin B1 production. So this is the gene cluster of aflatoxin B1 synthesis. And this is the early stage, middle stage, and late stage involved genes. So they checked the RNA-seq analysis results. So this that showed that genes involved in early and middle stage of enzymatic reactions of aflatoxins were significantly downregulated compared to wild type. So that means that when uridine filtrate was supplemented, the aflatoxin gene cluster expressed was down. And this AFLS is known to be a pathway-specific regulatory transcription factor, and this AFLS was inhibited under the treatment of Aspergillus uridine filtrates. These are the regulators of aflatoxin aflatoxin synthesis, and first you can see that that a negative regulator known NSDC is known as a negative regulator of aflatoxin production, and this was activated when uridine filtrate was supplemented. On the other hand, these genes are known to be positive regulator of aflatoxin production, and they were significantly decreased when uridine was supplemented to flavus. Then they wanted to know the effect of Aspergillus uridine filtrates on the expression of biosynthetic gene cluster, because previous results showed that uridine filtrate supplementation will regulate the metabolism in flavus. So they checked the BGCs in flavus, and they recognized that 13 transcription factors located in the predicted BGCs were analyzed, and 4 genes, among them 4 genes, were downregulated when uridine filtrate was supplemented. And most of the genes in the cluster of aflatoxin, this is aflatoxin and aspartic acid, and these are secondary metabolites of Aspergillus flavus, and these were transcriptionally reduced when uridine was supplemented. Also, both genes that are involved in leporin B, clavaric acid, and aflatoxin, which is also a secondary metabolite, were found to be depressed as well. So supplementation of uridine filtrates to Aspergillus flavus may play a role in expression of biosynthetic gene clusters. Nextly, they wanted to know the effect of Aspergillus uridine filtrates on the expression of biosynthetic gene clusters. So they checked the correlation within the genes. A total of 66 TF transcription factors were found to be expressed differentially by more than 2-fold, and among them 33 were upregulated and 33 genes were downregulated. And correlation heat map results showed that aflatoxin specific TF AF beta was negatively correlated with this gene, And another regulator, AFLS, showed a correlated expression on these genes. And among them, this bristle A and ABA A is known to be a central regulator of asexual development. So next, the researchers measured the researchers conducted a co-regulated gene expression network between aflatoxigenic and non-aflatoxigenic conditions. These aflatoxins have been shown to be regulated by many environmental features such as pH and humidity and temperature. So previous study reported that Aspergillus flavus failed to produce aflatoxin when grown in YFP media. So an interaction network analysis between the aflatoxigenic and non-aflatoxigenic conditions was analyzed. The left colors are modules, which is a cluster of genes that are correlated. So this color of turquoise and brown modules display the highest correlation with the aflatoxin phenotype. This one and this one. So the total of 325 interacting genes are involved in this turquoise module, and 87 genes of the brown module were involved. In the involved genes, 3 aflatoxin-related genes, these AFLH, AFLB, and HIBC, were downregulated in the non-aflatoxigenic conditions. Also, genes which were all downregulated in both Aspergillus oryzae filtrate treatment and YFP culture media were among the core regulated network. And the expression levels of genes involved in the TCA cycles and glycolysis were all significantly reduced in the non-aflatoxigenic conditions. That means the TCA cycle and glycolysis were potentially involved in aflatoxin biosynthesis. Lastly, the researchers wanted to know the inhibitory regulation of asexual development genes by the treatment of Aspergillus oryzae filtrates, because conjugation is one of the most important reproductive structures of flavus. So previous results showed that oryzae filtrates could suppress this asexual development of flavus. So to determine the inhibitory regulation of oryzae filtrates on flavus reproduction, the transcriptional conditions of genes involved in asexual development were analyzed. 2 central regulators of asexual development, as I formerly mentioned, bristle A and ABA A, were decreased significantly when Aspergillus oryzae was treated together. And the negative regulators of conjugation, such as these NSDC or VEA, were found remarkably increased in their expression in response to oryzae filtrates. This means that the negative effects on asexual reproduction involved in the regulation of positive and negative regulators of conjugation in response to Aspergillus oryzae filtrate treatment. So to summarize my presentation, the cell-free culture filtrate of non-aflatoxigenic Aspergillus strains can effectively inhibit the production of aflatoxin production and the growth of Aspergillus flavus. And the genes involved in the aflatoxin biosynthesis pathway and other BGCs were significantly downregulated. Lastly, 2 asexual development central regulators, this bristle A and ABA A, were found to be significantly downregulated, which might lead to the downregulation of conjugation. So this research provides new insights for the molecular mechanism of controlling aflatoxin synthesis to control the proliferation and pollution of Aspergillus flavus and aflatoxin. Thank you.